In this lecture, we will look at microbial growth and control. In this lecture, we will look at the way bacteria reproduce and the physiological conditions necessary for their growth. We will also look at terms such as asepsis, sterilise, disinfect, and sanitise, and understand the common methods used to control microbial growth, especially in the healthcare settings. We will understand the concept of sterile field and also look at ways to minimize the formation of aerosols. Physiological conditions for bacterial growth. Just like us, bacteria need the right temperature, the right pH, oxygen concentration, and also presence of food, water, salt, and mineral concentration for growth. Let's look at temperature as one of the physiological conditions for bacterial growth. Bacteria prefer to grow in an environment with optimum temperature. Optimum temperature is the best temperature for its growth. So the rate of growth will be the fastest in the optimum temperature. It can be classified into three broad categories based on the temperature at which they grow. For example, psychrophiles, they like to grow at cooler temperatures ranging from -20 to 10 degrees Celsius. And an example of psychrophiles would be the Flavobacterium species. Then we have the mesophiles. Mesophiles are those that grow in our body. Okay, so they are the most disease-causing forms of bacteria. They like to grow at temperatures between 20 to 40 degrees Celsius. You might know that our constant body temperature is around 37 degrees Celsius. So the bacteria in our body are usually mesophiles. Okay, for example, the E. coli and Staphylococcus aureus. Then we have the thermophiles. Thermophiles are bacteria that like to grow in very hot temperatures. For example, like the hot springs, thermal vents, and they grow at temperature range between 41 to 80 degrees Celsius. Would be the Bacillus stereothermophilus. As you can see from the graph here, okay, this graph has a rate of growth against temperature. Okay, you see that the blue line indicates the growth of psychrophiles, and the green one indicates the growth of mesophiles, and the red one or the pink one indicates the growth of thermophiles. So the optimum temperature is at the peak of its growth. pH is another important physiological condition for bacterial growth. Some bacteria, they like to grow in neutral pH around 7.0, and they are usually killed when exposed to acidic or alkaline environment. Stomach acids has a pH of less than 2. This is why most of the bacteria that you find in the food that you consume is killed in the stomach. Bacteria have their own optimum pH for growth, just like how they have an optimum temperature for growth. But you may be surprised that in an acidic environment like your stomach, you do have bacteria such as Helicobacter pylori that can survive in your stomach, and this is a bacteria that causes gastric ulcers. In the table below, you see examples of bacteria with their optimum pH for growth. For example, E. coli likes to grow the pH range of 6 to 7. Since this is close to a neutral pH, we call it a neutrophile. Okay, whereas Helicobacter pylori, it grows in an acidic condition, so we call it a acidophile. And then Bacillus alkalophilus grows at alkaline condition 10.5, so we call it a alkalifile. So in this graph, as you can see, the pH ranges from 1 to 14 actually, yeah. It shows up to 12, but the pH range is up to 14. So anything below 7 is acidic and anything above 7 is alkaline pH. Oxygen is another important physiological condition for bacterial growth. Here you see 5 tubes with different oxygen concentrations. The oxygen is the highest at the top of the tube and lowest at the bottom of the tube. So here you see Legionella bacteria growing at the top of the tube where the oxygen concentration is highest. This shows that this bacteria requires oxygen for growth. It can only grow where oxygen concentration is high. Such bacteria are known as obligate aerobes. Aerobes meaning they like oxygen. They grow in highly oxygen concentrated areas. Next, we look at Escherichia coli as an example of bacteria that likes to grow mostly at the top of the tube, but it also grows throughout the tube where there is less oxygen. Now, such bacteria, okay, they have greater growth in the presence of oxygen, but they are also able to grow where oxygen concentration is slightly lower. These kind of bacteria are known as facultative anaerobes. Okay? Anaerobes are bacteria that grow in the absence of oxygen. So since E. coli can grow in the absence of oxygen, as you can see at the bottom of the tube, we call them facultative anaerobes. Next, we have the obligate anaerobes. Obligate meaning they are true anaerobes. They only grow in the absence of oxygen. They are killed by the presence of oxygen. An example will be Clostridium tetani. Next, we have the aerotolerant anaerobes. So, aerotolerant meaning they are able to tolerate oxygen. Okay, but although they prefer the absence of oxygen, okay, they are only able to grow in anaerobic concentrations as because they do not use oxygen for growth, but they are able to tolerate the presence of oxygen, which means oxygen has no effect on their growth. An example will be your lactobacilli that is commonly found in your intestines. And then finally we have the microaerophiles, micro. Okay. They grow in environments with low oxygen concentration. They need oxygen, but they don't need too much of oxygen. So as you can see in this tube, they are growing in the middle of the tube because they just need low concentration of oxygen for growth. An example is your Helicobacter pylori that is found in the stomach of humans. Like any other living organism, bacteria also need the presence of food, water, salt or mineral concentration for growth. Bacteria need a basic source of food to survive, and this can range from simple sugars to a full course or complex meal in containing sugar, protein, and lipids. Bacteria and fungi also need water for growth, for survival. That is why dried fruits, which has no water or moist, can be kept for a very long time. Bacteria also need trace amounts of mineral salts and concentrations in small concentrations. For example, lactoferrin in the tears and saliva removes iron, and this is required for bacteria to grow. Without iron, bacteria is not able to grow effectively. So having looked at the conditions that bacteria need for survival, for growth, let's look at how bacteria reproduce. Okay. Bacteria reproduce by a process known as binary fission. Okay. So here, so the bacteria, when it's time for it to divide or to reproduce, it elongates and then it replicates its genetic material. This is the DNA of bacteria. It is now duplicated. Okay, now you have two copies. Then the cell membrane begins to divide like that. Okay, and each site will have a copy of the genetic material. So the cell membrane will completely form around the bacteria, and eventually the cells will separate into two different cells. Okay, here it shows a actual diagram of a bacteria dividing. So you can see there are two genetic material here, two copies of genetic material. The time taken for one bacteria, okay, to become two individual bacterial cells is known as the generation or doubling time. So this is one generation or doubling time. So if you start with one bacterial cell, let's say you start with one bacterial cell, after the first generation, you will have two bacterial cells. Second generation, you have four bacterial cells, and then it goes on to 8, 16, 32, 64, and so on. Okay, so this is exponential growth. Like that, exponential growth. You can calculate this exponential growth by using the formula 2 to the power n. So here 2 to the power n times initial number of bacteria. This refers to the number of bacteria that you start with. For example, if you start with 5 bacteria, okay, and then the 2 to the power n is your doubling time. So you'll be able to calculate the number of bacteria at the end of the the end of the process. Okay, so let's look at an example in the next slide here. Okay, so if you start with 5 bacteria, you have 5 bacteria now, and then they tell you that this bacteria has a generation time of 120 minutes, which means it only divides every 120 minutes. So what is the final number of bacteria after 2 hours? So you need to find out what is n. Okay, what is n? Okay, what is the generation? How many number of generations are there? Okay, so in 2 hours, how many minutes do you have? Okay, there are 120 minutes in 2 hours. So it's going to be 2 to the power 1, right? N is 1 because it takes 120 minutes to divide. So if we start with 5, so 2 to the power 1 times 5 cells. Okay, and so what will be our answer? It will be 10 bacterial cells after 2 hours. To the power 1 is 2 itself, right? So 2 times 5 is 10 bacterial cells after 2 hours. Look at another example here. Okay, here in the second example, you are given generation time of 40 minutes, and then you are told that after 2 hours you get 80 cells. So how many cells did you start with? So as you know, 2 hours is actually equals to 120 minutes, right? Okay, 120 minutes here. So if the bacteria takes 40 minutes to divide from parent cell to daughter cell, so how many 40 minutes are there in 120 minutes? The answer will be 2, right? Sorry, 3, right? So the formula will be 2 to the power 3 times your initial number is equals to 80. So now can you calculate what is the initial number of bacteria? So 2 to the power 3 is actually 8. Okay, so 80 divided by 8, so you get 10. So you started with 10 bacterial cells, which has a generation time of 40 minutes, and after 2 hours you get 80 cells. Hope you understand how to calculate generation time or how to calculate the number of bacteria after a certain period of time given the initial number of bacteria. Living with binary fission, not all bacteria have the same number of generation time. For example, Mycobacterium. This is the species that causes diseases like tuberculosis. It takes about 10 to 30 days to divide, which means one mycobacterium cell can take about 10 or 30 days depending on what kind of species you have, okay, to divide into two cells. Some bacteria like the Staphylococcus aureus and your E. coli they have very shorter generation time. For example, Staphylococcus aureus takes about 20 to 30 minutes to divide from a parent cell into two daughter cells. So like I said in the previous slide, this is exponential growth. 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, 32, 64, and so on. The graph here shows the phases in normal bacterial growth. Okay, so this shows a growth curve, bacterial growth curve. So this is the number of viable cells, okay, and here you have the hours in the x-axis. Now we start off with the lag phase. What is a lag phase? Okay, lag phase is where bacteria are trying to adapt to their new environment. Okay, so the bacterial population is quite stable, okay, and they are preparing to divide. They are preparing to adapt to the new environment, and then after that they will start to divide. In the exponential phase, over here you find the greatest rate of growth. Okay, so this is the active stage of bacterial growth. So bacteria numbers double with each generation time. Okay, so here 1 becomes 2, 2 becomes 4, and 4 becomes 8, and so on. Okay, and then it reaches a point, okay, where it goes into the stationary phase. So in the stationary phase, as you can see, the growth is what you call that is constant, which means the number of dead cells and the number of new cells are equal. Okay, so why does this happen? Because as you can see here, this thing blue here, okay, it shows that there is enough resources for the bacteria to grow at exponential rate. When it comes to stationary phase, you see that there are some dead cells, meaning, okay, the resources are depleting. There is not enough food, not enough uh, the optimum pH is not there. There's not enough oxygen probably because there's too much of bacterial growth. So they've already consumed most of the resources like food, oxygen, and all that. Okay. Then finally it comes to a point where it starts the death phase. This is where bacteria dies due to limited growth resources. As you can see here, you have more and more dead cells. Okay, so the number of dying cells exceeds the number of new cells formed. There are a lot of dead cells than new cells here. Okay, whereas in the exponential growth, you have a lot of new cells and very little or very few dead cells. This slide once again explains the different phases of the bacterial growth curve. Bacteria that are growing in the exponential phase are easy to be disrupted using antibiotics. Okay, it's easy to disrupt actively growing bacterial cells when they are in their exponential phase. Now let's look at how we can relate the phases of the bacterial growth curve to stages of disease and infection. Okay, let's say you visit a friend, okay, who has bacterial pharyngitis, a sore throat, for example, a sore throat. Now, when you visit this friend who has sore throat, it is very likely that you might get the bacteria from your friend. So, when you get this bacteria from a friend, you don't immediately fall sick. Okay, so, this is known as the incubation period, which means you have the bacteria in your system, in your throat now, okay, but you don't see any symptoms of the disease because the bacteria is adapting to the new environment. So, that is the lag phase. When the bacteria has adapted to your environment, it goes into the exponential phase where it actively divides, and then it will also be in the stationary phase after some time. This is when you see the clinical symptoms and signs of sore throat. So you have throat pain, difficulty in swallowing, and all that. You might even have a mild fever. So then what happens? You visit a doctor. Okay. So the doctor prescribes some antibiotics or some kind of medication, and then you start to recover. Okay. So recovery is the convalescent stage, and this is actually the phase of decline for the bacteria, the death phase in the bacterial growth curve. We're going to look at how we can control bacterial growth. Okay, because we've looked at what bacteria need for growth with physiological conditions, and then we looked at how bacteria divide. Okay, now we look at how we can control the growth of bacteria. And before that, we will look at bacteria that can form spores. Okay. Many gram-positive bacteria, they can sporulate, okay, which means they can form endospores. Okay, they can form endospores. What are endospores? Okay, so let's look at this diagram first. You see, this is the vegetative cell. Vegetative cell means actively growing bacteria. And when the conditions for growth are very harsh, for example, very high temperature or very low temperature, okay, the pH is not optimum, there is not enough food or water. So what happens? These gram-positive bacteria, okay, they develop spores here. See these spores? Internally, they form spores, and then eventually they become free endospores. Now, endospores are resistant to heating, drying, freezing, radiation, chemicals, okay, and many things that would kill a vegetative cell. So, vegetative cell means cell that is actively growing. So, when these bacteria become endospores, they cannot be killed easily. Okay, and what happens? These endospores, when they find that the environment is suitable for growth, when there is enough oxygen, enough water, the right pH, then they become vegetative cells again. Okay, so sterilization methods such as autoclaving can be used to kill these bacteria. But if you were to use just soap or boiling, okay, it will not be able to kill the or get rid of the endospores. Before we proceed any further, let's look at some common terms that will be used when we discuss the next few slides. Okay, so sanitize, sanitize means to mechanically remove microorganisms. For example, during your hand washing, when you use soap, or when you clean your eating utensils, you are actually sanitizing them. We call it sanitize. Okay, next we have disinfection. Disinfection destroys all vegetative pathogens. Okay, so like I said, vegetative refers to actively growing microorganisms. Okay, so when you disinfect with chlorhexidine or by boiling, okay, you are actually destroying all actively growing microorganisms, but you cannot get rid of endospores through disinfection, or endospores and viruses cannot be destroyed through disinfection methods. Next we have sterilization. To sterilize means to destroy all or remove or destroy all microorganisms, including endospores and viruses. So if you use these sterilization methods like gamma radiation, autoclaving, ethylene oxide gassing, you will be able to remove all microorganisms, even endospores and viruses. Next, we look at asepsis. Asepsis refers to the practice or that prevents the entry of pathogens into sterile tissues, and this prevents infections. Okay. An example will be using iodine to prepare skin for surgery. You remove the bacteria that's on the skin before an incision is made during surgery. Another example would be the use of alcohol swab before an injection is given to a patient. So you use the alcohol swab to remove the bacteria or the any microorganisms on the skin, surface of the skin, before you give an injection. Otherwise, when you give an injection, the bacteria or the microorganisms will be able to enter the skin as well. This chart shows you some methods and techniques for the control of microbial growth. For example, you have the physical agents. Okay, they can be heat or radiation. So, for example, hot air oven can be used to sterilize equipments. And then, if you have any contaminated or used syringes and needles, they can be incinerated for disposal. Then you have the autoclave machine that can be used for sterilization. This is also heat, moist heat. And then you have the boiling water. Now, boiling water is not a sterilization method, it is a disinfection method. Recall from the previous slide, disinfection methods do not destroy endospores. They also do not destroy viruses. They only destroy actively growing cells, the vegetative cells. Physical agents, okay, can also be radiation in the form of gamma radiation, which is a sterilization method. So it will destroy all microorganisms. Or it can be a disinfection method such as the ultraviolet or UV light. Next, we have the chemical agents. Okay, gas, we have ethylene oxide. Okay, it can be used for sterilization. It gets rid of all microorganisms, including endospores and viruses. Okay, chemical agents, liquid chemical agents. We have the hydrogen peroxide at a high concentration. It can be used for sterilization. And then we have the alcohol and chlorhexidine, okay, which are disinfection methods. For mechanical means, we have the filtration methods. HEPA filter, okay, is used to sterilize air. So in the operating theaters, you'll find HEPA filters that will sterilize the air. And then if you are using liquids, if you want to what do you call that, sterilize liquids, you use membrane filters. Autoclave is a moist heat sterilization method that can be used to sterilize equipments. So here these two pictures show you the autoclave. So if you want to sterilize equipment, okay, you place them, or instruments, you place them into this drum, okay, and then the autoclave will operate under pressure of 15 pounds per square inch at a temperature of 121 degrees Celsius, and you only need 15 minutes for sterilization. So this autoclave is the standard instrument for preparing things like the microbial media. This is the liquid or media that you need for bacteria to grow. Okay, your agar plates, all these are microbial media. And it can also be used to sterilize instruments such as your syringes, okay, your hospital garb, okay, your lab coats and all that, your blankets, and also IV solutions in bottles. Okay, these can be sterilized using autoclave. So after autoclaving, the packaging will have a expiry date, usually 1 month from the date of sterilization. Another example of sterilization method would be the ethylene oxide gas. So ethylene oxide is normally used to sterilize objects that are sensitive to temperatures or radiation. So things like your suction tubing, okay, or hand pieces, catheters, like these are catheters here. Okay, radiographic film holders like this, radiographic film holders. We want to take an X-ray of your teeth. Okay, all these cannot be autoclaved because they are made up of heat-sensitive material like plastic. So how do you sterilize these instruments or materials? We use ethylene oxide gas. So this is a cabinet where you load all the things that for what you call sterilization by ethylene oxide. and then you expose these instruments to 2 to 3 hours of ethylene oxide gas, and then your equipment becomes sterilized. You should know that ethylene oxide gas is toxic, carcinogenic, which means it can cause cancer, and it's also flammable. So we only use these when the objects to be sterilized are sensitive to temperatures or radiation, which means they cannot be sterilized using gamma radiation. For liquids, if we want to sterilize liquids, okay, we use membrane filters like this, membrane filters. Okay. So we use a syringe and then attach a membrane filter to the syringe, and then you can sterilize the liquids through this membrane filter. So solutions that are used for dialysis, okay, so intravenous therapy or surgical irrigation, they contain substances, okay, in the liquid that can be affected by high temperatures. So to sterilize them, we use membrane filters. So other heat sensitive solutions include drugs, okay, vitamins, culture media meaning media that is used for bacterial growth that cannot be autoclaved because it will destroy the proteins inside the media. So these solutions can be sterilized using membrane filtration. Yeah. Okay, air can be filtered using HEPA filters. Okay, HEPA filters are used in operating theaters, burn wards, okay, isolation units, and also communicable disease wards. So here is an example of a biosafety cabinet that you might find in the microbiology laboratory, which also uses HEPA filters. Okay, so the HEPA filter will remove any bacteria that is in the environment. Okay, so we removed. Filter will trap any bacteria, okay, before the air is let out into the open. So the air that leaves the cabinet is filtered and is therefore clean. We all harbor microbes, which means there are microorganisms on our body, on our clothing, on our pencil case or bags. Okay, so we can easily contaminate our environment as well. Okay, we can contaminate our own experiments in the lab and also the sterile field. Sterile field means an area where it is free of microorganisms, which you can use to work with microorganisms, other microorganisms. Now, we also must be mindful that we can be infected with microorganisms as well. Okay, so these can also affect others working nearby. It can also affect your friends when you leave the, let's say, the microbiology laboratory without washing your hands. You may be carrying some microorganisms and you may transfer it to your friends who are not in the lab with you. Hence, it is very important to exercise good aseptic techniques. Okay, an important point in aseptic techniques is the concept of non-touch procedures. Okay, you might know that surgeons, okay, in the operating theaters, after washing their hands, they usually keep their hands above their waist level, right? They usually have their hands up in the air, okay, because they do not want to touch anything else and contaminate their hands again. If you want to use any equipment in the operating theater, they must be sterilized before every operation. Okay, and then you must ensure that the plastic or combustible material are not flamed for sterilization, because flaming them or using heat to sterilize them will destroy them. So probably you should use gamma radiation techniques like gamma radiation or ethylene oxide gassing. You can see here in this picture, okay, the equipment, the scissors is being opened without touching the scissors. This is the non-touch procedure. So all sterile packaging are sterile on the inside but not on the outside, because you can see bacteria on the outside here, right? So before you open any sterile pack, such as this pack of cotton buds, okay, you should perform hand washing because you do not want to transfer bacteria from your hands to the package. You must also check the expiry date. Okay, like I said, after autoclaving, the materials are only sterile for about a month. There's an expiry date. Check the expiry date. Check packing is not damp. Okay, if it is wet, it is very easy for bacteria to have entered the package. Make sure the seal is not broken. You see the seal here, okay, at both ends of the bacteria of the package. Make sure they are not broken. Check the sterile field is already established, which means you have a place which is clean, which has been swabbed with alcohol, which is free from any microorganisms. Okay. Check that there is no draft. Draft means current of air in an enclosed area. For example, if you are working near the door, okay, when someone opens the door, okay, there is an exchange of air between the air inside the room and air outside the room. So it can bring in what do you call that, microorganisms into your working area or your sterile field. And then finally, open at the handle end of instruments. Open at the handle end of instruments. Do not open at the top of the package here where you have the cotton buds. Okay, always open at the handle end of instruments. Especially when someone sneezes, you find very fine particles, okay, in the air, and these carry bacteria and viruses and other microorganisms. Also contaminate your sterile field or your what do you call that, experiments. So in the lab or in the microbiology lab, we can also cause aerosols without sneezing. For example, formation of bubbles from liquid microbial cultures. When you're given let's say a tube of bacteria, do not shake it, okay, forming aerosols or forming bubbles, because when these bubbles escape, okay, when these bubbles escape and pop, they release microorganisms into the air. So avoid shaking your cultures vigorously, okay, when dispensing using droppers. Okay, also avoid bubble formation. So we've come to the end of the lecture. We have understood how bacteria reproduce, what is it they need for their growth, for example, optimum temperature, optimum pH, the right oxygen concentration, and also the presence of food, water, and mineral salts. We also learned the terms used in healthcare, such as sanitise, disinfect, asepsis, and sterilise. We looked at the common methods used to control microbial growth, and we also understood the importance of integrity in the sterile field. Okay, how important it is to maintain a sterile field when working with equipments that are sterilized or working with equipments used for operations. Okay, and how we can minimize the formation of aerosols. I hope you have understood the lecture. Okay, see you at the next lecture. Thank you.